All right, so we're going to go over the 2013 AP Biology FRQs that I gave you guys in a packet last week. Yeah, you're close to the camera. You're being recorded.
Already?
Yeah. All right. So, question 1. In an investigation of fruit fly behavior, a covered choice chamber is used to test whether the spatial distribution of flies is affected by the presence of a substance placed at one end of the chamber. To test the fly's preference for glucose, 60 flies are introduced in here, so that's an important point to keep in mind. 60 flies. Into the middle of the choice chamber, so they're inserted right here. And a cotton ball soaked with a 10% glucose solution, like so, is placed at one end of the chamber, and a dry cotton ball with no solution is placed at the other end. The positions of the flies are observed and recorded for every 10 minutes. Every minute for 10 minutes. So for A, we're supposed to predict the distribution of flies in the chamber after 10 minutes and justify it. What do you guys think? Where will the flies be located?
Yeah.
So, most of the flies not pleased, will be in the glucose chamber. Why? So, because they are attracted to sugar? Yeah. Sorry. I know it is. Maybe I'll end up on some weird teacher blooper channel. Because they are attracted to sugar. Yes? So, from what I know, mosquitoes are attracted to CO2 that's released when you breathe out. Since all animals respire and breathe out CO2, they're attracted to that. And that's the way they can find a food source. But there may be some other chemicals that are attracting them as well. All right. All right, so you get 2 points for this. 1 point for predicting the location and 1 point for justifying that prediction. I want to go back and point out the fact that if you remember, anything that's in bold indicates that's the way to earn a point. You have 2 bold words. Each of them is asking you to do one thing. So you have a maximum of 2 points that you can earn for this. This is, also remember, questions 1 and 2 are 22-minute long questions. It's recommended you spend 22 minutes on them, and they're worth 10 points each. So those are the ones that are the biggest point value. They are worth half of the points you can possibly earn on the free response. So B says, propose one specific improvement to each of the following parts of the experimental design and explain how the modification will affect the experiment. So what is the experimental control in this setup as we have it?
The cotton, yeah.
Which cotton?
The glucose and the dry, both.
There is only one control, the dry cotton.
Oh.
Our variable that we've introduced is the glucose. So let me ask you, you have glucose-soaked cotton and you have dry cotton. Are those 2 pieces of cotton different from each other? How many ways?
Only one way.
Well, one of them has glucose solution because it's got a 10% glucose solution. It's soaked. The other one is dry. So over here, you have plus glucose and you have plus water. Over here, you have minus glucose and you have minus water. If you have any difference in the choice that the fruit flies make, can you tell whether it's due to glucose or due to moisture? No. You have 2 variables here that you're comparing. You need to only have one thing different between those 2 conditions. So how could I modify my experimental control?
Miss, how do you know there's water though? If it says glucose soaked, it doesn't say glucose water soaked.
It's in a 10% glucose solution. All of our solutions in biology are going to be in a water solution unless they tell you otherwise, because everything that happens in our bodies is in a watery or aqueous solution.
So if it were to say 100% glucose, it'd be different?
Well, 100% glucose would just be glucose powder. It has to be dissolved in some water. Make sense?
Yes.
All right, so what can we do to this control to make it a more valid experiment?
Add water to the dry cotton.
So for the control, we can add plain water to the,
You have to just add water, you can't add nothing else.
because if you add anything else, that's going to be another difference. Like let's say you added sucrose. Well, you don't know if they're attracted to glucose versus sucrose. First, you want to tell whether they're attracted to, it's always best to have a negative control, because if they're equally attracted to moist cotton that's moistened with just water and sugary moist cotton, then if they're equally attracted, you know that the smell of the sugar is not doing anything different. But for the sucrose, you can't, if you had sucrose on one and glucose on the other, you can't say, oh, their preference is for the molecule. It could be due to just the fact that they're interested in the water. So you need to eliminate as many variables as possible. All right, so environmental factors. Are there any environmental factors that we could manipulate?
It's hot. Is weather count like hot or cold?
So, not 100%. That might be a control, because that's not like your control group, that's your controlled variables. So all of your other factors, including temperature, humidity, etc., should be controlled. What about in place of, is there anything in place of the glucose-soaked cotton that you can think of? Because think about it, is there pure glucose just sitting out there in nature?
Yes.
Not really.
Or
Usually,
not pure.
not pure, and also generally not glucose, it's table sugar.
What about like a leaf?
A lot.
Do fruit flies eat leaves?
No. No, but
What do you
I
think
mean,
fruit flies eat?
Fruit.
Fruit. So instead of glucose-soaked cotton, what could we do?
Make a piece of
fruit-filled cotton.
fruit-filled cotton.
Like take the juices of a fruit and put it on a piece of cotton.
So instead of glucose, the cotton could be soaked in fruit juice. Bloody hell.
Yeah.
Oh, that's something.
Apple juice.
Y'all was laughing at me and I was right.
I wasn't laughing at you.
I
I was
was.
laughing at what you said.
I was laughing at you.
Whoa. I don't know what this thing is doing anymore. It is possessed.
Technical difficulties just for the video. I gotta narrate because, you know,
You should be a director.
narrate.
His voice isn't that good though.
Ooh.
And yours is?
I'm not saying mine is.
I mean, I could change my voice.
Go.
You were like, oh, this is being captured on video.
I don't even care at this point. Right, the video is ruined. We might as well just do I'm too far into
edit, so.
Just
yeah,
post it.
this video is already
Alright.
messed up.
So, this
Y'all
part
didn't
was
scumbag
worth
me.
4
Now we're at
points.
sunset and I was like,
You get 1 point for your proposed improvement to the experimental control, and a point for explaining it. You could also get 1 point for the environmental factor versus also getting 1 point for the explanation. That totally threw me off. All right, so you do need to make sure that if you're, for example, saying that you're going to replace the dry cotton ball with a water-soaked cotton ball, you need to make sure that you have this explanation. It ensures that glucose is the attractant. Michael said make sure that your temperature and your environment are constant. That would be another thing that would have counted as experimental control. And your explanation is because all other variables need to be held constant. For environmental factors, you could use, you could basically perform variations on the basic experiment they had. So different concentrations of glucose, different temperatures. So that actually gets into what Michael said. Different light levels, a different size or shape of choice chamber. You could do the duration of this experiment instead of for 10 minutes, do it for 10 hours and check once an hour. Or you could vary the time of day. So the way that would be refinement is you can tell if you add any of these other variables and the results are the same as
If I don't wash it.
just your original variable, you can say that the flies are only moving due to a glucose preference. Keep in mind the scoring guide that I have up here is what the College Board releases, but at the time, oh, that disappeared, okay. At the time that they're grading these, they do take alternate explanations as long as they're correct.
I never said that.
What?
We're talking about this.
He said that if you use pineapple juice, it will work better. I said no.
Actually, I think bananas are their happy place.
Yeah.
All right. So, C, the experiment described above is repeated with ripe bananas at
See,
one
miss,
end.
this is where. Oh.
Okay, so hold on, we're gonna go over.
Calm down, Gil.
And unripe bananas at the other end. Once again, the positions of the flies are observed and recorded for every minute for 10 minutes. The positions of the flies after 1 minute and after 10 minutes are shown. Perform a chi-square test on the data for the 10-minute time point. We need to specify the null hypothesis and enter the values from your calculations. So first, let's do a null hypothesis. Null hypothesis usually states there's no relationship between our variables, so we will not see any effect due to our variable. So our null hypothesis should be something along the lines of the fruit flies will be evenly distributed.
See, I don't even know what chi-square. I forgot.
It's a square.
No, it's not.
Will be evenly distributed amongst the 3 locations. So our null hypothesis that we want to either find support for, or we want to either reject it or fail to reject it, is that the fruit flies will be evenly distributed everywhere. So how many fruit flies do I have total?
60.
I have 60 fruit flies total. So that's going to be in my observed data. For my observed data, the number of fruit flies at 10 minutes that are at the end with the ripe banana is 45. In the middle, 3, and at the unripe end, 12. Now, keep in mind that our expected number has to be the same, because we are expecting out of those 60 flies, how many would we have, or how many would we expect in each chamber? There are 3 places they could be. So there are 3 places they could be. We expect them to be evenly spread out.
20.
20, 20, 20.
Oh yeah, look, there goes. There's the answer.
Quiet
I
you.
saw it.
Quiet me or quiet him?
Him or
Quiet
him who?
everyone. All right, this next part requires a calculation. So I'm not going to do it in that box because it's not going to fit. They expect you to do your calculations elsewhere. So it's your observed minus your expected, that value squared, divided by your
What
expected.
is 25 squared?
So it's 25 squared
600.
divided by
10,000.
20, and that is 625, right?
Yes.
Jeanette, what was the value you got?
She's small.
I like calculus.
31.3, I'm going to round it.
Calculus could say the same for you.
For the next value, it's going to be 3 minus 20
No,
squared
kid Michael. I didn't like
divided by 20. And Jeanette, what did you get for that?
25.
So I'm going to round it to 14.5. And then the last one, 12 minus 20 quantity squared divided by 20. And what did you get for that? And when you add them all together, you got
You gotta dab me up on that one.
48.9. So, altogether, this earns you 2 points. Or, yeah. 2 points. So you needed to fill everything in, and you needed to have a null hypothesis that says they'll be evenly spread out.
I didn't actually answer that.
I have money.
Any questions?
It's in my book.
No, man.
So,
I got a dog.
these are your various restrictions. These need to be values that are predicted by the null hypothesis. Everything needs to add up to 60, and no cells can be filled with the number 0. All right, then we need to explain whether our hypothesis is supported by the chi-square test and justify our explanation. At this point, it is nice to have your formula chart out because your formula chart has the critical values table.
My mom just got a job to pay $60.
Realize you're still on the internet. So our chi-squared value was about 48, let's say.
I'm gonna be on YouTube.
How many different locations could we have fruit flies?
3.
3. That means our degrees of freedom is going to be our number of possible outcomes, which is 3, minus 1. So our degrees of freedom is 2. For a p-value of 0.05 on your critical value table, so p-value of 0.05 on your critical value table with degrees of freedom of 2, your critical value is 5.99.
Okay, can I ask a question?
Yes.
Hi, my name is Lester, and I would very much like to know what is degrees of freedom?
Degrees of freedom. It's you calculate it how I told you. It basically allows you to have a factor of the number of different possible outcomes you can have.
Oh my Jesus.
Yeah,
Jesus
that would
Christ.
get into more statistics than I am prepared to go over right now.
But what's the purpose?
The purpose of this is to see, could our results be due to chance, or are they the result of some other variable? So wait till we get to the very end of this, and we'll talk about that again.
So the higher the degrees of freedom,
The more possible outcomes you have, the bigger your critical value can be. Because if you look at it, your calculations right here are the result of the difference between your observed and your expected, but they're weighted by your expected value. If your expected value is like 100, then having a difference of 25 is not going to be that big a deal. But if your expected value is 10, having a difference of 25 is going to be a bigger deal. Then you're adding together the result of looking at the difference between your observed and your expected, what you saw and what you expected to see, and you total all that up. And then if it's lower than a certain value, you know that, okay, we can fail to reject our null hypothesis, so you can put a little more stock in the null hypothesis.
Okay.
If the number is bigger, like our chi-squared value was 48. Our critical value is 5.99. Is our chi-squared value bigger or smaller than our critical value? It's bigger. So because of that, we reject our null hypothesis. So in this, they do say that you need to clarify that these are the degrees of freedom, this is the p-value, and that the critical value is 5.99. You compare them, and you decide whether or not the null hypothesis should be rejected, or you fail to reject it. In this case, we reject it, and because of that, what we can say is, the fruit flies are not evenly distributed. Something else must be causing the difference in their distribution. So instead of them being evenly split up, they are clumped at one end or another. And that is a statistical test to help validate the fact that you're saying, oh, these fruit flies are attracted to the banana side. That's what the support for this is. So then, the next thing, we need to propose a model that describes how environmental cues affect the behavior of the flies. So how do we get environmental information into our bodies?
Eat it.
No, how do we get information about the outside world into our bodies?
Go outside.
I don't know what he's talking
You
about.
go outside.
How do you know what I'm saying?
Oh,
We hear
it's
it?
because it's.
I don't even want to answer that, bro.
How do I know what you guys look like? Where
You're
you
looking
sit
at us.
You gotta
I
test
get
it.
input. I get sensory input.
Sensory.
I was gonna say sex, but
What do you think that the fruit flies are using?
Their senses.
Which sense?
The fifth. The
They're
fifth.
probably
the
using
fifth
their
sense,
olfactory sense, which is their sense of smell. And then what happens to that sensory information?
Fucking got hoes right here talking about make it smell.
Make it smell?
Where does that sensory information go?
To the brain.
brain.
The little brain.
They do have a
Smart.
tiny little brain in there.
Penis.
you step on it, they'll die.
Yes.
Step on them, they'll die.
Not every living thing.
They kill them flies.
Higher animals, definitely all vertebrates have brains, and some invertebrates do too, like squid and octopodi and all of them.
I beat a bee's head off.
Insects
A
have
bee
multiple
pooped on my
brains.
face one
They're
time.
just tiny.
Yeah, you're disgusting.
Okay,
That
so
was
once
Lester
information
Walt.
goes to the brain, what happens to it?
You're disgusting, bro. You better be.
Dude, you just said you.
Okay, so then it makes a decision. It's supposed to be makes. About how to respond.
That's like an S. Is it percent? Is it square?
Ew.
Or is
Did
that
it
square?
again. Possessed.
It's racist.
It's processed.
It's racist.
Alright, so that is actually what earns you a point, that you have a stimulus and then you have a response. Or you can say you have an input, like a sensory input. You make a decision or you process it. That's integration of information. And then you have an output. Questions, comments, concerns? Alright, let's look at number 2. Do you guys want to go over number 2 since we've already gone over it in class?
Number 2. Oh, that was number 1?
All of that was number 1. This is a 22-minute long question.
How long did it take us?
I don't know. Y'all should have been looking at a watch.
It's 22
Let's-
minutes that it took us.
Let's
That is
skip
exactly
2
22
and let's go to
minutes.
3. We've already gone over 2 in class, so I don't want to spend time on it. So fossils of lobe-finned fishes, which are ancestors of amphibians, are found in rocks. They're at least 380 million years old. Fossils of the oldest amphibian-like vertebrate animals with true legs and lungs are found in rocks that are approximately 363 million years old. So when we're looking at rock layers, which layers are older? The ones on the top or the
One
ones
on the bottom.
The ones on the bottom.
So this would be 380 million years ago. And these are lobe-finned fishes.
What the hell? What was that?
All.
What was that?
That was like some Doctor Strange into different dimensions. Can I delete that I said that?
Alright,
I just saw that
363
movie.
million years old would be above that, and that would be the oldest vertebrates, oldest amphibians. 3 samples of rock are available that might contain fossils of a transitional species between lobe-finned fishes and amphibians. We have 1 rock sample that's up here. It's
Where'd it go?
I don't know. Gilbert, stop doing your magic.
It's because you know
Your Doctor Strange
your
to
computer
you.
knows you're teaching a black person, so they don't want me to gain any knowledge.
Some street knowledge.
Wow.
Street knowledge. Michael said that. That was Michael Wagner.
Wagner.
How do you say it?
Wagers.
Michael Wagers.
This video.
Why is he wearing a suit?
So up here, that's
Not a suit.
350 million years ago. We have another sample that's
That's a fit.
370 million
fitting.
years ago.
It's not
And we have
fitting.
another
It's
one.
tight.
Look at he got the Gucci bag.
Okay.
It is
I feel
Just
bad for this
completely,
video. It's over with.
everything disappeared.
This video done.
350, 370.
You want to be where the funny
390.
people were at?
Which one of those, it says select the most appropriate sample of rocks in which to search for a transitional species and justify your selection. How many points do we think this is worth?
5.
2.
2, because we got 2 bolded words. Alright, so where do you think we're going to find a transitional species? We have 380 million year old rocks have lobe-finned fishes. The oldest fossils of our amphibians are 363 million years old.
Well, in order to predict that, first we must know what is a transitional species.
So what is a
species that
transition?
Because
transitions.
I have no idea.
Like it changes.
Transition in a species.
A transition, like a transition in a paragraph, if you're writing it, would be like then, or next, or secondly. It's something that moves you from one place to the next.
It's obviously locked.
On the other hand, in this,
Stupid.
a transitional species would be a species
She
that...
left.
See,
Who was that?
Just sit there, never mind.
Anyway,
I'll be like, well, you then.
hey, you're on...
They don't know what I said. Man, at this point, whatever
This
said
video's
is just, you
canceled.
should just let it be. This video been ruined like
You just
Twitter.
stamp it with the PG.
Shazam it.
It's Leslie said because your
Rated
computer
R.
knows you're teaching a black freak.
I know.
She
By the
recording.
way, Lester thinks everything's racist.
What if she
so-
get like 10 million views?
I would watch this.
The
It's funny.
funniest biology class ever.
You're like those.
Hey, so sad.
Okay.
You're like those teachers who US history put like the shows.
Yeah,
I like one of those teachers.
Oh, Lord.
That
Oh.
everybody wishes. Oh, I wish I had her class.
I hope so. Otherwise,
You messed it up, coon.
Knock.
You got 140 today?
Yes.
Do people not know how to knock anymore? You go like
No,
that.
I don't.
That's how you do. She's gonna.
Hey, you know, knocking's in style.
Hey, Diane, you know how to knock?
Hey, stop messing up the video, okay? We're messing it up more.
That's
All right.
I love you.